everyone po. Kamusta po ang araw natin? Thank you po sa pagdalo sa ating webinar ngayong araw na ito. Once again, good morning everyone and welcome to our webinar series entitled Livestock Biotechnology Center Research Development and Extension Agenda Technical Caucus Webinar Series. My name is John Louis P. Baligad, and I am the moderator for today's webinar session. Thank you very much for making the time to join with us today. Now, our webinar series is initiated by the Livestock Biotechnology Center together with the Philippine Carabao Center. This online activity aims to disseminate innovative livestock biotechnology research results with appropriate policies, advocacies, for a competitive, profitable, and sustainable livestock industry, even amidst the pandemic. Now, the first part of this webinar series aims to provide an overview of the RDE agenda of the DA Biotech Program Office and research presentation on genetic diversity. So, before we begin, I'd like to share a few reminders. First, please be informed that this webinar is being recorded, and while the speakers are in session, you may send your comments and questions to the chat box. And please remember to indicate to whom you're addressing the question to. Also, in asking, you may introduce yourself, and at the end of the webinar series, we will be sending a post-webinar assessment form via email. And after you send us back the post-webinar assessment form, we will be sending a certificate of participation to all the attendees. So ready na po ba tayo? Ready na po ba ang bawat isa? And I think everyone is ready to listen and learn from the two speakers that are experts in their field of interest. But before that, let us hear first from Dr. Claro N. Mingala, the Chief of the Livestock Biotechnology Center. Yes, Doc.
Narinig niyo ako. Okay. Good morning again. I am very glad to see all of you and our participants for today. The sudden and unexpected coronavirus or COVID-19 has had a drastic impact on the way people work in just a few months. It tends to boost the development and adoption of new technologies in a changing and challenging environment. Webinars such as these, virtual meetings, and video conferences have been of great assistance to continuously support events without eliminating safety measures meant for the speakers and participants. Moreover, this pandemic correspondingly has given impact on the livestock industry as operational sustainability of food supply chains is crucial in preventing food crisis and alleviating unfavorable results on the global economy. Through the technological advances in communication, limit on the spread of information has been raised and become faster and wider. The Livestock Biotechnology Center, along with the Philippine Carabao Center, intends to unceasingly disseminate latest livestock biotechnology research information in the country by initiating various technical caucuses, focusing on breed improvement, livestock product development, animal health, and nutrition. This is to strengthen innovative research results with appropriate policies and advocacies for competitive, profitable, and sustainable livestock industry. Thank you very much for participating, and may this activity be beneficial to everyone. Maraming salamat po.
And thank you, Dr. Mingala, for your warm welcome. Now, let us proceed to our first speaker this morning. Our first speaker is currently the Project Management Officer of the DA Biotech Program Office. which is in charge of the implementation of the program's biotech research and development agenda. She will be discussing briefly the role of biotechnology to the society and its current implications. Now, ladies and gentlemen, please join me in welcoming Ms. Marie Joy Christine Humalon.
And thank you for inviting us in this activity. As already mentioned, I am the Project Management Officer of the DA Biotech Program Office. And in behalf of our Director Coordinator, Ms. Annalyn Lopez, she sends her regards to you. She's in another engagement right now because she's holding two offices. Besides her stint in our office, she's also part of the Policy Research Service of the Department of Agriculture. So they're very busy right now. But I hope to do justice in this presentation. Okay, so I will present on the program's RDE agenda and advocacies focusing on livestock biotechnology. So everything that we do in the department, kindly click the next slide, please, are hinged on the three major global challenges. So we all know that population would continue to grow exponentially while our agricultural land will continue to decline. And with that, we experience or there's a perennial problem on attaining and sustaining food security at viable levels of sufficiency consistent with national security and sovereignty. So food security, according to the United Nations, is not just availability of food, but food being available to everyone at any time and for all people of any economic status. So hindi lang siya available but also accessible for everyone and affordable for everyone. We are also facing this increasingly distorted international market, so we need to keep up with for our agricultural commodities to keep up with competitiveness. And of course, as we all know, we are also experiencing the challenges of climate change. So with this in mind, the DA Biotech program is crafting its R&D agenda to help address these principal challenges. Okay, so why biotechnology? So biotechnology is just one of the options of the department of our government to address the mentioned challenges. But why did they invest or why are they investing in biotechnology? Well, for one, biotechnology expands the toolbox for breeding and disease management. Some techniques help or complement and make conventional methods more effective. Like for example, the use of gene sequences and molecular markers reduces the time for breeding, whether it be for crops or for animals. And these are also very useful tools for our regulatory agencies in terms of authentication or accurate identification of species. Some of the techniques of biotechnology also overcomes the limitations of conventional methods. Like for example, for breeding, if we compare conventional breeding with advances in biotechnology tools, which they call now precision breeding, kindly proceed to the next slide, please. Okay, so compared to conventional breeding, our breeding tools in biotechnology allows the development of traits even from distantly related organisms. With conventional breeding, we are limited to traits that can be developed resulting to exchanges between the same or very closely related species. And this normally takes a long time to achieve because you undergo trial and error in breeding. But with precision breeding, as the name or as it is coined, desired traits can be achieved in a shorter period of time because you limit your choices and you know your target traits and you know how to generate it using your knowledge of the DNA. Okay, so the DA Biotech Program Office is a result of the 1997 Agriculture and Fisheries Modernization Act. So the or what they call AFMA. AFMA prioritized biotechnology and allotted specific budget, allocated budget for the biotech program. So in 2000, the Department of Agriculture created the Biotech Program Office. So the Biotech Program aims to contribute to the general or the main mission of the department to move the Philippine agriculture and fisheries sector from resource-based to technology-based. And the main purpose of our office is to help orchestrate an enabling environment so that we can use wisely biotechnology as one of the options to improve productivity, attain and sustain food security, as well as improve resiliency to climate change. Next slide, please. Okay, so how does the biotech program address the challenges of Philippine agriculture? So we do this by supporting studies and activities classified into four areas. Okay, so sabi kanina, the purpose is to help create an enabling environment for the use of biotech. So first, we aim to generate improved biotech products and processes under the biotech research and development component of the program. So it's the typical support to research and development so that we have the technology that can be useful for our beneficiaries, mainly the farmers and our regulatory agencies. Okay, so in order to help create the environment of adoption, the program also has three other components. And these are policy research and advocacy. Under this component, we support projects or studies and activities that would guide the decision-making of our policy makers and decision makers in the government for the use or to help in the utilization or adoption of biotech products. Okay, so another component is the institutional capacity enhancement, wherein we help research institutions and regulatory agencies in increasing their capabilities to generate and manage biotech products and technology. So because as we all know, some of the biotech products might have regulatory implications or may need to undergo regulations. Okay, so another component of the program is the information, education, and communication component. So parang kumbaga balawala yung mga technologies na ma-generate under biotech research and development kung hindi naman siya gagamitin ng ating target beneficiaries. And some of the products of biotech needed to be communicated to the general public to facilitate the adoption of these products nga. So yun, this component aims to increase public awareness and understanding on biotech and its applications. Okay, so these are some of the result of our support in this four components, specifically for policy research and advocacy. So since continuous ang advancement in technology and there are policy needs for some of them, we continually support the formulation and continuous strengthening of science-based and functional regulatory system. So one of our, sorry for that, my daughter is calling me. So we help create the enabling policy environment for biotech, but continually supporting our regulatory agencies in developing and strengthening science-based and functional regulatory system. So isa po sa sinuportahan natin ay ang pag-finalize ng Joint Department Circular number 1 series of 2016, which is the basis for regulating or assessing the biosafety of genetically modified crops. And so now we are also supporting the current efforts of the Bureau of Animal Industry together with the other key regulatory agencies. And this is actually being spearheaded by Dr. Clara Mingala. So they're trying to come up, they are trying to come up with guidelines for regulating or assessing the safety of genetically modified and genome edited animal products. Yan po. So tuloy-tuloy po yung support natin dyan. Under the same component, or PRA, or Policy Research and Advocacy, we also support studies that would guide our R&D investment in the program. And so, for example, if there are advances in technology, for example po, itong mga supported studies na to on genetic modification, like for example, genetic modification, GM abaca technology. So wala pa po tayong GM abaca in the Philippines, but we've supported the conduct of this study that would analyze the feasibility or marketability of this technology if ever. So yan po yung mga sinusuportahan natin under this component. We also supported the establishment of the 3 agri-biotech centers. Yung organizer po natin today, the Livestock Biotech Center, is part of that. We also have our Crop Biotech Center hosted at PhilRice, and Fisheries Biotech Center hosted at NFRDI. The goal of the centers are to help advance the research and development using biotech tools. Yan. Okay, so I already mentioned earlier yung about information, education, and communication component of the program. So we are a regular organizer of the annual National Biotech Week celebration. So this activity aims to increase public awareness and understanding on biotech. And we have initiated or pioneered several activities along with the celebration. So we initiated the honoring of the Filipino faces of biotechnology kasi yan, yung mga nakikita nyo po sa slide, these are the key people who helped or played a key role in advancing the biotechnology applications in our country. So yan po ang ating, kumbaga, star when it comes to biotechnology. So these are farmers, scientists, policymakers, people from the media who helped promote and advance or facilitate the advancement of biotechnology in our country. And we biotech crops versus zombies, we funded the creation of this application para ma-appreciate yung ating mga biotech crops and so on. Hello? Next slide, please. Yan. Under institutional capacity enhancement po, we support upgrading of laboratories. Yan. So both in research and development institutions and in regulatory agencies. So we grant lab equipment to upgrade nga the capacity of the laboratories, and we also so that to capacitate research institutions to continue to conduct biotech research, and for regulatory agencies to be able to perform their mandate related to regulating products of biotechnology. So let's focus on the RDE agenda on livestock biotechnology of the program. So the overall direction of the biotech RDE agenda is to help enhance the resiliency and quality of agri-fishery commodities, as well as ensure sustainability of the industry. So the research theme or focus can be classified or can be grouped into 3 research themes. First, we aim to produce animal health biotechnologies such as disease diagnostic protocols and kits and vaccine. Our priority diseases are those of the priority of the Bureau of Animal Industry. We also aim to develop breeds that would have enhanced production, reproduction, product merit, and adaptation traits. And we also want to develop animal nutrition biotechnologies, such as improved breeds or immunobiologics or probiotics and prebiotics. Okay, so for now, up to 2022, our priority commodities are water buffalo, cattle, goat, swine, and chicken. And the Bureau of Agricultural Research, being also a government funding agency under the same department as ours, actually there are overlaps on our priority research themes, but they focus on research on native animals. So that's how we complement or harmonize our efforts on research and development. Next slide, please. So bakit tayo may ganitong focus on our RDE agenda? Well, for one, if you would see on your screen, the self-sufficiency ratio on most of the livestock commodities is less than 100, which means we are still not self-sufficient on these commodities, except for chicken egg. Excuse me. Now, because of the presence of diseases such as African swine fever, this further is reduced. Okay, so for example, for pork, it is predicted that by this year, mag-lessen ng 10% itong ating self-sufficiency ratio. So this is just a reflection of all the challenges that the livestock industry is facing. Okay, so ang daming challenges, no? So lack of availability of quality stocks of animals, insufficient or unstable supply of feeds, presence of many diseases, which significantly lowers our domestic production, climatic disturbances, poor management practices, adaptability problems of animals, meat safety and quality, environmental degradation. So this is an issue of sustainability of the industry, slow transfer of technology, long lead-in period and high investment, lack of knowledge, technical knowledge of farmers. Maybe meron pa kayong ibang naisip, but these are main challenges of the livestock industry. So the self-sufficiency ratio is just a reflection of these challenges. So admittedly, we cannot address all these challenges, especially those that are outside the mandate of the DA Biotech Program Office. But we also have the livestock program in the department, and the livestock program of the Department of Agriculture aims to address the other challenges of the whole value chain, because the DA Biotech program is focused on addressing the diseases. We aim to help address the technical aspect, or those that can be addressed by biotech research and development only. But even if we are limited by our mandate and our resources, because unlike other programs, the biotech program, kung iko-compare mo siya with other government funding agencies, we have relatively we have fewer resources than them. So we want to make sure that we generate solutions that would be very relevant to our target beneficiaries, which are directly or indirectly the farmers and even researchers and even our regulatory agencies. So let me quote from Jane Hart. She says that people will only want to engage if it is relevant to their needs or helps them solve problems. So we want to be relevant to our target beneficiaries. And let me also quote our director coordinator, Annalyn Lopez. She said that no mountain is too high when everyone is determined to climb. So we started with the global challenges of Philippine agriculture. I know parang, 'di ba, food security might be an ongoing problem hanggang, 'di ba, parang it might not be very possible to attain food security in the near future. But yun nga, if everyone is willing to contribute to solving these problems, it's gonna be easier, or it's not gonna be impossible. So we at the DA Biotech program, we are committed to creating meaningful partnerships so that all of us can work together in attaining our goals, in terms of securing every Filipino, securing that every Filipino that they would have enough food, accessible and affordable food for them. So for those of you who we haven't worked with or partnered with, we hope to work with you in the future, and we hope that you also help us create meaningful research programs for the DA Biotech program, because at this stage po, so we presented to you the RDE agenda, but we are still refining this. We are creating specific research programs that would help implement this agenda, and we hope that you can help us create meaningful and relevant research programs in order to implement this target research and target output. Yun lang po, maraming salamat. Good morning, everyone.
Yun, thank you, Ms. Joy Humalon, for your very informative presentation on the RDE agenda and advocacies focusing on livestock biotechnology, specifically the role of livestock biotechnology to the society in and its current implications. And personally, I love this quote from Jane Hart. Sabi, people will only want to engage if it is relevant to their needs or help them to solve problems. So naway makapaggawa tayo ng mga biotechnology researches na mas makakatulong sa ating mga farmers at para po makatulong din tayo sa food security po natin in the future. Now, we would like to acknowledge po yung attendance ng ating mga newly tuned in na participants from different research institutions, academies, and sa lahat po ng mga PCC centers mula dito sa headquarters and sa mga PCC centers around the Philippines. Thank you so much po for making the time to join with us today. And thank you so much sa mga participants po natin from Pisao, Pampanga, VSU Leyte, DARFO2, RADDL, kamusta po dyan? Doc Tin Mamawag, Doc Roxanne, at sa lahat po ng mga bagets. Sa UP, thank you so much po, UP. And Primsu, kay Sir Sani na nakatutok ngayon, thank you so much po, sir. And of course, DABPO. And sa lahat po ng nakatune in po ngayon dito, thank you so much po. Now, bago po natin simulan yung ating second presentation, before we proceed to the second presentation, in today's webinar session, as I am said earlier, you may send your comments and questions to the chat box, and please remember to indicate to whom you're addressing the question to. And we will having our open forum after the second presentation. Our second presenter is a graduate of Bachelor of Science in Biology at Brigham Young University in Hawaii. She took her master's degree in Marine Science at University of the Philippines, Diliman, Quezon City, and her PhD ongoing in Bioscience at Tokyo University of Agriculture in Atsugi, Kanagawa, Japan. A recipient of various local and international awards and recognitions, she is currently a senior science research specialist and the current head of Cryobank unit here at the Philippine Carabao Center National Headquarters and Chinpool. Ladies and gentlemen, we will now give the screen to Ma'am Lilian Villamor.
Okay, sorry. So thank you very much. Good morning, everyone. Louis, thank you for the introduction, and also thank you for the Livestock Biotech program, Livestock Biotechnology Center, for giving me the opportunity to share with you the funded project of the DA Biotech entitled Genetic Diversity of the Philippine Carabao using Mitochondrial DNA COI and Microsatellite Markers recommended by the FAO. And this project started in 2015 and will be completed this year. And this project will not be possible without the support and the contribution of the project leaders and project researchers. I'm William Moore. I'm the head of the cryobank unit, the project leader of this study. And I work with Dr. Esther B. Flores, who is the head of the Animal Breeding and Genomics section of the Philippine Carabao Center. And her contribution is focused on the morphological characterization of Philippine swamp buffaloes. We also work with Dr. Annabelle Sarabia, the RDE chief. And during the initiation of this project, she was the chief of the research division. And now she's enjoying her life as a retired government employee. I also work with dedicated young researchers, and one of those is Alexander M. Paraguas, and his contribution is on the phylogeny and maternal lineages of Philippine swamp buffaloes using mitochondrial DNA markers. Another is Ivy Joy Escuadro, and her work focused on population structure and genetic diversity Philippine swamp buffaloes using the microsatellites, or also known as the short tandem repeats. And Therese Patrica Kailipan, she was highly engaged on the identification of male crossbred buffaloes using the SRY gene, COI, and SDR markers. To start with, in Asian countries, Bubalus bubalis, or water buffalo, is an effective source in the agriculture economy and food security. With significant variations, water buffalo was categorized into major types: the swamp type and the river type. So they are morphologically different. Swamp type has more white or light gray markings that could be found on his feet, what we call the white stockings, also the chevron, and the horn is crescent shaped that is different from the riverine buffalo, which has curled horn shape, and the color pattern of the riverine are darker compared with the swamp type. So what have been the conservation efforts in the Philippines? In 2003, an open nucleus of 50 cowhead of swamp buffaloes was established in the Philippine Carabao Center at Cagayan State University. Village-based gene pools had been identified by PCC at CSU in Tabuk, Kalinga, with 25 heads of cows dispersed together with a bull. The island municipality of Carlos P. Garcia, also known as the Putogo Island in Bohol, had been identified by PCC at Ubay Stock Farm as its island sanctuary of swamp buffaloes. So these are part of the ex situ conservation of the buffaloes across the Philippines. And the Philippine Carabao Center also supports actually these are the in situ, I should say, and the ex situ conservation through cryobanking has been also supported by the Philippine Carabao Center. So what's the history of domestication of Bubalus bubalus in the Philippines? Following the domestication in China, the domesticated swamp buffalo spread through separate routes. The first one is through Taiwan and the Philippines to the eastern islands of Borneo and Sulawesi south. And the second one is through mainland Southeast Asia, and then to the western islands of Indonesia. So let me just share with you previous studies that have been done locally about the diversity of swamp buffaloes in the Philippines. This is an article lifted from the main author, which is Dr. Cacho, and it's entitled Genetic Diversity Analysis of Water Buffaloes Populations in the Philippines using the bovine microsatellite markers. And for this study, she used 10 microsatellites, and one of the findings was the Philippine Carabao, which is the swamp buffalo in our country, is separated from the riverine buffaloes. Another study by Del Barrio et al. Using the mitochondrial D-loop sequences, the results the result reported the differentiation or detection of lineages A and B. But for this study, the samples only fell into the lineage A and none in lineage B. A genetic study and relationship of domestic buffalo and cattle breeds and crossbred in the Philippines based on the COI, the cytochrome C oxidase gene sequence, was reported by Dr. Orville Bondoc. And one of the findings is that among swamp-type Philippine carabaos, no significant geographical structuring was found, perhaps due to greater and repeated gene flow and extensive transportation along local carabao populations caused by inter-provincial and inter-island accessibility. So the big question, or the long-standing question of this study would answer, is there really a genetic diversity on the Philippine carabao? So answers to this question imply two possible directions toward the in situ conservation of the species. First, if the answer is no, meaning there is lack of genetic diversity in Philippine carabao subpopulations, this status of gene diversity entails that the present institutional conservation of Philippine carabao at Cagayan State University will be sufficient to represent the genetic traits of Philippine carabao in the country. But if the answer is yes, that there is a genetic diversity in Philippine carabao subpopulations, the genetic diversity will define the rationale of strengthening the current conservation, but not limited to expand the strategic conservation and management of the Philippine carabao in the country. To answer this, with the focus of the general objective is to support the effort of developing genetically improved products of livestock through the use of highly polymorphic markers that have the capability to identify and discriminate breeds of native carabaos in the Philippines. And these are identified in three specific objectives. Number one is to determine the haplotype structure of the native carabaos in the Philippines using the DNA barcoding mtDNA COI. Second is to assess the genetic diversity of swamp buffaloes based on a set of polymorphic markers like the microsatellite or STR markers as recommended by Food and Agriculture Organization. And the third objective is to evaluate the usefulness of the microsatellite markers for conservation management and future breeding of these species based on these genotypes. So this slide is showing you the sampling sites that are collected across the islands of Luzon, Visayas, and Mindanao. So in Luzon, 13 sampling sites were collected, 7 in Visayas and 8 in Mindanao. So a total of 26 provinces across the Philippines. So the next pictures I'm showing you is showing you the topography of the landscape topography of the sampling sites. So you can imagine how isolated and challenging the transportation for these islands. Good enough, there are swamp buffaloes existing over these isolated islands and mountainous places. So for instance, in San Luis Aurora, Itogon, Benguet, it is only representative samples from Samar, Pitogo Island, and also in Cagayan, Calayan Island in Cagayan. So moving forward, the methodology consisted of the blood collection from the swamp buffaloes. And as part of it is we interviewed the farmers to ensure that these animals that were included in the study would be unrelated. And the blood collection was also isolated using the Promega commercial kit. PCR amplification was done for the mitochondrial markers as well as the STR markers. And for the analysis of the mitochondrial DNA, we focus on the sequence alignment and the statistical analysis, actually for the mitochondrial, for the sequence alignment and the phylogenetic analysis. And same methodology except for the data analysis using the microsatellites. It's more on the allele scoring and the population structure. Also included the genetic variation as well as assess the genetic differentiation. And since we're very interested in the genetic information, but we also took advantage of gathering the morphological characterization. So this is just showing you how we consider data gathering for the morphological characterization that focus on the morphometrics for the body, horn, and face measurements. And the second one is more on the physical characteristics, including the fur color, skin color, as well as the pattern and others. So for results and discussion. So focusing on the result and discussion to answer the first objective of this study, we infer the phylogeny and network structure of COI sequences from the Philippines, and this shows a separation between the swamp and the riverine. And also we detected two haplotypes in here. We initially call it the minor haplotype, and majority of the haplotypes for the other group. The second figure showed the median joining network of PC and swamp buffaloes retrieved from NCBI. And focusing on the maternal lineages in the swamp buffaloes, this marker was able to separate the riverine with the swamp type, and we also detected swamp lineages using the NCBI markers. But focusing on the haplotypes that we detected in COI from the Philippine samples, it wasn't able to give us a better resolution to delineate the two lineages. And this explains further for the third picture of the COI that only two haplotypes with a one base mutation was determined using the COI. So the COI is a good gene marker, but it is limited to confirm the species identification of Bubalus bubalis as swamp type or river type. And COI has very conservative for this swamp buffalo population analysis. And this is consistent with the result of DNA barcoding reported by Bondoc 2013. So we were not satisfied with the result of the COI. So we investigated our MTDE markers. We thought about there got to be genetic diversity. So we're not convinced for the result of COI. So we moved to other mitochondrial DNA, and we started with the 16S ribosomal RNA. And this one buffalo is a little bit conservative, but it was able to detect the second lineage, which is the maternal lineage B, which was detected in the OCM sample or the Occidental Mindoro. However, few samples fell in line B for this analysis. So in this 16S, line A stands as a dominant haplotype, wherein most samples were incorporated in this lineage. So we moved to a different marker, which is the cytochrome B, really investigating the genetic diversity of the mitochondrial maternal lineages of the swamp buffaloes. And this is showing that the clustering of the swamp buffaloes is consistent with the results showed by COI, with the major haplotype and the minor haplotype. And this marker also showed the separation with the riverine buffaloes with the swamp type. And focusing on the swamp population, cytochrome B revealed the two lineages A and B, and major haplotypes are considered in lineage A and few haplotypes in the lineage B. So the mitochondrial work is getting more excited, but we're not yet satisfied. So we want to determine more of the genetic diversity using the mitochondrial DNA. So in here, we incorporated the two regions of the mitochondrial D-loop sequences for the shorter, which is 216 base pair, and the bigger one is, or the larger one is the 365 base pair. So let me talk about the 211 base pair of the mitochondrial DNA. So we were happy about this result because it gives us a better resolution of the delineation of the swamp lineage A and the swamp lineage B. And focusing in the lineage A, it's major haplotypes fell into this lineage, and few of the samples detected from Caucus, South Cotabato, and Sultan Kudarat, I should say, and South Cotabato were detected in this lineage B, and actually even the Occidental Mindoro. So the other region of the empty DNA D loop also supported this analysis, but similar to other mitochondrial, it was able to separate the riverine with the swamp buffaloes. And now we'd like to focus on the mitochondrial maternal lineages. And we're happy enough to detect the lineage A and lineage B of these swamp buffalo populations in the Philippines. So this is just giving us a summary on the information of various empty DNA markers that we investigated. Just to recap, we started with the COI, but because of very low haplotype number and nucleotide diversity and one variable site, it would not be enough to discriminate the maternal lineages of A and B that was previously reported in the swamp buffaloes. And this showed the increasing pattern or resolution to elucidate the genetic diversity of Philippine carabao based on increasing number of haplotype from COI to mitochondrial D loop, the nucleotide diversity, as well as the variable sites. So since we found more information on the mitochondrial D loop and the site B, we focus on these mitochondrial DNA markers to interpret the maternal lineages diversity in the Philippine carabao swamp populations. And as we determine the information on the mitochondrial DNA, we also would like to know the paternal identification of these animals. So when you will see the picture of this animal, it exhibits features of swamp buffaloes with the presence of horn shape, crescent shape, and with the presence of chevron and white stockings. But if you're in doubt of the integrity of this breed, genetic marker can be able to establish not only for the maternal, but also for the Y chromosome or the paternal identity. So the big question, is this really a true swamp type buffalo? This animal was tested in mitochondrial, and the mother is swamp. But let's see if the father is swamp as well. Using the SRY gene, which is a Y chromosome, it showed the nucleotide polymorphism, a change from C in river to G to swamp. And this gene is telling us that this sample is a putative cross, is a crossbred, because the mother is swamp but the father is riverine. So this marker has been useful for us, was useful for us to eliminate the samples that are putative crossbreds. But this is only limited for the male buffaloes. So to answer the second objective, assessing the genetic diversity of swamp buffaloes using the microsatellite markers, we determined the genetic diversity parameters across the Philippine swamp buffalo populations. And this is just showing you a few of these parameters. We have 30. We started with the 30 microsatellites markers, but they have different behaviors. So we focus on the presence of the polymorphism, which is accounted on the polymorphism information content. And these markers, 17 of them, are highly polymorphic, which means that the PIC value is greater than or equal to 0.5. 9 of these STR markers are reasonably informative with a value of 0.30 to 0.49. And one loci is slightly informative with less than 0.20. And we also found monomorphic markers. So with this analysis, we remove this monomorphic because it will be important for the genetic diversity analysis. This is showing you the summary of the global F statistics of the 26 Swamp buffalo subpopulations. For the FIS, it is showing the overall significant deficit of heterozygotes. FST is more on the genetic differentiation, and the FIT is the global deficit of heterozygote across populations. So we use the classification of the genetic differentiation based on the FST value was based on the Wright's classification that was reported in 1978, which means that greater than 0.05, it means actually less than 0.05 means little genetic differentiation, and moderate genetic differentiation is above 0.05 but less than 0.15, and great genetic differentiation is above between 0.15 to 0.25, and about 0.25 it's more on very great genetic differentiation. So the Philippines, actually, the overall FST or genetic differentiation among the 26 subpopulations was 20, actually it was 20.6, actually 0.066 or 6%. And that is significantly different. For the analysis of AMOVA, among populations, there is a percentage variation of 6.61. Among individuals, among populations, 7.84. And the greater percentage variation was found within individuals of 85%. We determined the population-specific FST. So what the question is, what is the genetic differentiation within the population? So the population FST value ranges from 0.061, the highest in Occidental Mindoro, and the lowest is 0.072 in Gigantes Island, indicating moderate genetic differentiation within the individuals among the populations. So now is showing the differentiation of the populations with other populations. So this is the matrix of significant FSTP values of the subpopulations. And we just want to highlight on those populations that have highest or 100% significant FST values between the 25 populations. So these are the populations in Kalinga, Carabao Island, Occidental Mindoro, and Gigantes Island. We also found FST value significant that is 96% different from the other populations, indicating genetic differentiation in Calayan Island, Camarines Sur, Batanes, and Capiz. So with that genetic differentiation, we found out that Calayan versus the Cagayan, with the Carabao Island, Gigantes Island, and Pitogo Island, they have a high genetic differentiation between these two populations, or actually within these two groups. So based on the NACE genetic distance, Cagayan and Gigantes Island and Iloilo were the most distant populations, while Sutan Kudrat and Sarangani were the closest distant. So this is just showing you the NACE genetic distance compared with the pairwise, the phylogenetic relationship. And we just want to highlight that the relationship of the populations of the swamp buffaloes across the Philippines are geographically consistent. So the populations in Luzon clustered together, as well as with the Visayas populations and Mindanao. This is a figure for the three-dimensional visualization of the principal component analysis of 26 swamp buffalo subpopulations, showing the distribution of individual samples across Luzon, Visayas, and Mindanao. And we just want to show that the individuals among the populations in Visayas and Mindanao are closely clustered, or they highly closer with these populations. But compared with the Luzon, they are kind of scattered. So this is the PCA as well among the 26 subpopulations. And it's consistent that even within the populations, Luzons are differentiated, genetically differentiated, but the Visayas populations and Mindanao are clustered. And looking closely, if you can, the Carabao Island, which is geographically located in Luzon, has higher affinity to those populations in Visayas. And basically, we were able to find an information that based this affinity of the Carabao Island to the Visayas population can be explained by the proximity and accessibility to neighboring populations in Visayas, and can rise from the history which entails changing assignment of the Carabao from Panay Island to Luzon population in Romblon. Okay, moving forward, to evaluate the usefulness of the microsatellite markers for conservation and future breeding of this species based on these genotypes, we performed the structure analysis. And this is showing what is if there is a population structure and what is the optimal structure of the population. So they show that structure analysis at K3 and K4, that Swamp 1 cluster for Calayan Island and Swamp 2 cluster for Carabao Island, Gigantes Island, and Pitogo Island still merge as distinct clusters. And one thing common about them is that they are isolated islands. The travel time varies from 30 minutes to 5 hours by boat from the mainland to the island. And the physical barrier caused by great bodies of water could impede genetic exchanges between the neighboring subpopulations, which most likely resulted in isolation of population by distance. We also have the average morphometric values of the swamp buffaloes according to the structure analysis. So with the Swamp 1, it's referring to the cluster in Calayan, Swamp 2 for the cluster in Visayas, and Swamp 3 is referring to the admixture of these two clusters. And noted that for the body length, the Swamp 2 is significantly higher than those in Swamp 1 and the Swamp 3. However, Swamp 1 has favorable height, heart girth, neck circumference, face length, and horn length, as well as the horn circumference and distance between the tip of horns compared with the other swamp cluster. So for the highlights of this study, the efforts on morphological and phylogenetic analysis of the Calayan Island helped the Department of Agriculture, the PCC, as well as the centers of the LGUs to identify Calayan as the other native Carabao sanctuary in Luzon. Information dissemination was done in Calayan Island, Pitogo, as well as in Carabao Island for these distinct subpopulations. We also shared this knowledge that for molecular analysis for species identification and genetic diversity with various researchers, students on animal biotechnology. And to summarize, the presence of genetic diversity and population structure of swamp buffaloes were identified across Luzon, Visayas, and Mindanao. So going back to the long-standing question, if there is diversity, these research findings confirm that indeed there is genetic diversity of the Philippine swamp buffaloes in the Philippines. Two maternal lineages A and B in the Philippine Carabao were detected based on DNA sequences using mtDNA markers. New lineage B was detected and traced in 6 Philippine Carabao populations, including Occidental Mindoro, Caucus, South Cotabato, and Sultan Kudarat. Distinct clusters of pure type swamp were detected in Swamp 1 cluster from Palayan Island in Cagayan, and Swamp 2 cluster from Carabao Island in Romblon, Gigantes Island in Iloilo, and Pitogo Island in Bohol. And based on the structure analysis, these animals have membership coefficient of above 95% to be a member of these clusters. Population structure in clusters of swamp populations reveal congruity of morphometrics and DNA profiles. For recommendation, these identified pure type Philippine Carabao in Palayan, Pitogo, and Carabao Islands can be considered for in situ and ex situ genetic conservation. Identified subpopulations of pure type swamp buffaloes in the country can be a take-off point to understand the economic traits for growth, rate, meat quality, and adaptation. And that will be possible if we were able to generate and establish the whole genome sequences of the Philippine Carabao of the swamp type in the Philippines. Because as of now, we don't have the genome sequences of this species. And this is the most important part of our slide, is to acknowledge the funding support that we obtained from Department of Agriculture, the Livestock Biotechnology Program, the Livestock Biotech, the Bureau of Agriculture, Agricultural Research, and the Philippine Carabao Center as the implementing agency. We also highly appreciate the local farmers and the different PCC centers across Luzon, Visayas, and Mindanao. And of course, the LGU, Vivait, and the regional staff. And this is showing you how we were able to, for our sampling sites and our joint efforts with the local government agency and the PCC regional centers, Luzon, Visayas, and Mindanao. And with that, thank you very much for listening.
Thank you, Ma'am Lilian, for the very informative presentation on genetic diversity of the Philippine Carabao using mtDNA and microsatellite markers, and the importance of these kinds of research on the breed improvement of the Philippine Carabao, and to support the effort of developing genetically improved products of livestock. Once again, thank you, Ma'am Lilian. Now, let us proceed to the open forum. You can now ask questions to our speakers. So po pwede na po tayong magtanong. And since nasa open forum na po tayo, pwede na po natin buksan yung ating mga webcams. Okay, so muli, gusto po namin pasalamatan yung ating mga speakers and participants from different government research institutions around the Philippines. Yung ating mga partner academe din po. Thank you so much for joining us today. And pwede po tayong magtanong ng live. Kung meron po kayong gustong itanong, pwedeng i-raise nyo lang po yung hand. Meron po tayo dyan sa emoticon. I-raise nyo po yung hand and then i-highlights po namin kayo for your question po. So, it looks like we have a first question from Anonymous. Ma'am Joy Humalon. So, ito po yung question para sa inyo. Ang tanong po dito, Ma'am Joy, sa tingin nyo po, nasaan na daw po yung ating implementation ng livestock biotechnology researches dito sa ating bansa? Hello, Ma'am Joy.
Hello. Can I ask for further clarification of the
Yes po, ma'am.
question? Kumbaga, ano na ang status ng
Yes, the status po, ma'am.
ng livestock biotech research in the Philippines?
Yes po, ma'am.
Okay, I'm not very technical, pero based on our consultations before, especially when we were crafting the RDE agenda. Actually, when it comes to livestock, we are sobrang behind pa tayo when it comes to utilization of biotech tools sa livestock research. Kasi parang ang main pa rin na ginagamit ngayon, the usual or the traditional tools like artificial insemination, and then the research of Miss Dr. Lilian Villamor is one of parang mga ganitong level pa lang yung ating na pondohan din sa research. So mga genetic diversity analysis, genetic molecular marker identification. But we also funded, ang isa sa mga napondohan namin kay PCC before with Dr. Paragison Alili, if I remember correctly, ay yung production ng detection for foot and mouth disease. So, pero other than that, parang wala pang masyadong progress when it comes to production ng vaccine. More on detection protocols ng mga diseases pa lang yung meron tayo. But we hope to help advance the research in or more yung use ng biotech tools sa livestock research. More on mga identification, molecular markers, ganyan po yung ating mga alam na mga napopondohan pa lang using in livestock biotech research. Yeah, we also have several projects on markers for determining the reproductive or fertility of livestock products under PCC rin. So mga ganong level pa lang po. Pero wala pa tayo dong, unlike sa crops na merong genetic modification, genome editing, wala pa po tayong ganon na mga kind ng research dito sa Philippines sa pagkakaalam po natin.
Yes, ma'am. Thank you, Ma'am Joy Humalon po sa answer po sa ating unang questions. And from sa answer po ni Ma'am Joy, so maganda po siguro encourage natin yung ating sarili or yung ating mga research institutions or academe na gumawa pa ng mga mas sabihin na po nating mas mataas po na antas ng research para po mas makapag-contribute po tayo sa livestock biotechnology aspect po.
Can I comment a bit pa? So,
Yes,
we
ma'am.
understand
Okay
naman po the current capacity rin ng ating research kasi when you venture into biotech research, talagang malaking investment 'yan. Hindi lang sa equipment pero syempre lalo na sa livestock parang sobrang daming expertise, napaka-diverse. So actually one of the challenges din naman kaya rin I think when it comes to pag-advance ng research natin or ng RDE agenda, specifically ng DA Biotech program, ay yung kakulangan ng experts. Well, I'm very happy to see na ang daming participants, pero hopefully talagang magtuloy-tuloy sa career sa research ang mga kumbaga next in line natin. So yeah.
Yes, hello Ma'am Joy, may follow-up question po si Anonymous. Pwede daw po ba silang gumawa po ng proposal, concept proposal, and then pupuntahan po siya directly ni DABPO?
Definitely, especially if it's in line with the priorities of the program. So yung diniscuss ko po na RDE agenda kanina, actually siya until 2022, but most likely it would still be the same theme pag in-update yung ating RDE agenda kasi ang dami po talagang pwedeng gawin dito sa research themes na to at saka actually we haven't progressed so much in implementing it kasi we have problems with kukonti yung nagpro-propose sa amin, so we actually encourage your submission of proposals. Kaya lang po ang aming budget ay fully allocated until 2021. So proposals that would come in would most likely be funded 2022 pa po at the earliest. Yun na po yung pinakamaaga na mapondohan yung mga papasok na proposals.
So yun po. Thank you, Ma'am Joy. Habang hinihintay po natin yung additional fund for new project proposals, so pwede na po tayong gumawa ng ating proposal para po pag meron ng available funds si DA Biotech, ay po pwede na po tayong mag-submit. Okay po. So any questions po?
Yeah, we encourage submission kasi matagal din naman ang review process. So, hindi naman ibig sabihin 2022 pa tayo mag-submit ng proposal. So, pwede na rin po kayo mag-submit ng proposal next year. Yan po, for consideration. And every time naman po kasi may mag-free kasi po nakaplano po, may naka-allocate na sa projects ongoing and anticipated to complete their review process. Sometimes may mga na-free up po na pondo. So we still entertain talaga yung mga incoming proposals. So kung meron na po kayo dyan, naka-ready, submit nyo na po. Pwede na rin.
Okay, thank you, Ma'am Joy. So it looks like we have new questions from John William Gamis. So this question is for Ma'am Lilian. Is there any plans for cryopreservation of gametes to genetically diverse buffaloes?
Yes, definitely. With this basic information that based on the genetic information that these distinct subpopulations will be the top priority for future ex situ conservation through cryobank.
Unmute po pala ako. So meron po ulit a question from Sir Joel Salazar. And this question is for Ma'am Lilian again. So with the result of your study, Ma'am Lilian, is there a specific gene or genes that indicate to increase the milk produce of Carabao?
Unfortunately, we were not able to include the markers that will answer that gene on milk production. So that's the limitation of the study. It's basically more on the identification of the diversity in the mitochondrial, the Y chromosome, as well as the nuclear genes for the parental genetic information.
Thank you, Ma'am Lilian and Sir Joel Salazar. And any question po from the webinar room?
Permission to ask po.
Okay po, sige po, Sir Joel.
Joel Salazar po from Pampanga State Agricultural University. Opo, para po sana kay Ma'am Lilian.
Okay, yes.
Yes,
Yes,
hello
sir.
ma'am, good morning po.
Good morning.
Opo,
Doc.
hindi ko po kayo na-meet po nung klase namin sa inyo. Last time po. Wala po
okay
ata
po.
kayo na, no? Anyway po. Ma'am, ask ko lang po,
Yes.
doon sa mga previous studies niyo po, meron po bang na-research kayo about doon sa kung paano natin may increase yung milk production ng mga carabaos natin, or specific lang kailangan silang macross from, let's say, sa mga buffaloes?
Okay. Sorry, Sir Joel Salazar.
Yes,
Regarding for the milk production, milk traits, that is beyond the scope of my study. Basically, with my 5-year stay in the Philippine Carabao Center, my studies focus mainly on the cryobanking, for conservation. So,
okay.
sorry, I might not be the right person to answer that.
Okay po.
Sorry.
Pasensya na pa.
Hindi, okay lang.
Yes, hello po. Thank you, Sir Joel and Ma'am Lilian. Any question po? Marami pa po tayong oras para po sa inyong mga tanong. So, kung pwede po tayo, wag po kayong mahihiya. Sa DA Biotech Program Office po, nandyan pa po si Ma'am Joy. Baka may mga gusto pa po kayong information na malaman regarding po sa kanilang agendas and advocacies po, pwede na po nating itanong. Okay po, so it looks like we've covered all your questions po. So sa mga tanong pa po natin na hindi pa po natin naiisip ngayon, po pwede nyo pong isend sa amin, and then itatry po namin ibigay sa inyo yung kasagutan through sa mga naging speaker din po natin. So ibibigay po namin sa inyo yung aming email address. So doon nyo po pwedeng i-email sa amin yung mga additional questions kung may mga maisip po kayo right after the presentations. O pwede po namin siyang sagutin at ibigay po sa inyo yung reply po ng ating mga expert. Okay po, so once again po, thank you so much, Ma'am Lilian, Ma'am Joy, and sa lahat po ng ating mga participants. And we couldn't have done this successfully without our participants and of course the expertise of our speakers. So let us hear what's inside this certificate. So present this certificate of appreciation to Ms. Marie Joy Christine Humalon for the time and expertise as a resource speaker during the Livestock Biotechnology Center Research Development and Extension Agenda Technical Caucus Webinar Series signed by Dr. Claro N. Mingala and Dr. Yoprosina P. Atabay, the RDE Chief of the Philippine Carabao Center. And also, this certificate of appreciation is presented to Ma'am Lilian Villamor for the time and expertise as a resource speaker during the Livestock Biotechnology Center Research Development and Extension Agenda Technical Caucus Webinar Series, signed Dr. Claro N. Mingala, the Chief of Livestock Biotechnology Center, and Dr. Yoprosina P. Atabay, the OIC Research and Development Division, Philippine Carabao Center. And for this certificate of participation, para po sa ating mga participants ay bukas po namin ipipresent sa inyo because meron pa po tayong second day sa ating webinar series tomorrow. And once again, thank you everyone for participating. Yeah. Once again, thank you everyone for participating this morning webinar session. We will be inviting you again for tomorrow's webinar. The presentations will be about breeding selection and repro biotechnology. So, may continuation po ito. Natapos na po natin ngayon yung genetic diversity as well as the DA Biotechnology Program Office RDE Agenda and Advocacy. So, tomorrow, we will be talking about the breeding selection and repro biotechnology. So now we have come to the end session, and I would like to invite all of us for the photographic sessions, and please open your webcam. Okay, so po paki-open po at nang makita po yung ating mga nagwagwapuhan at naggagandahang participants around the Philippines. Ayan, so where your killer, ayan, smile, killer smile. Okay, and 1, 2, 3. Okay, so once again po, thank you so much po for attending this webinar. And once you leave today's webinar, you will receive a survey, a post-assessment form, and we would appreciate if you would complete that and provide your feedback. So, kapalit po nung pag-fill up nyo ng aming post-assessment form ay yung doon po namin i-base yung certificate of participation nyo. So please, paki-fill up po siya ng buo para po sa ating, especially po yung inyong family name. Dapat full name po ilagay po natin doon para po sa pag-encode po ng inyong certification. Okay. On behalf of Livestock Biotechnology Center and the Philippine Carabao Center and our presenters, thanks again for joining us today, and we will see you tomorrow. Maraming salamat po, and God bless Pilipinas.